Good morning, students. Welcome to the biology class. We are continuing with our chapter cell cycle and cell division. In our previous session, we discussed about the cell cycle, the phases of cell cycle, that is interphase and M phase. Today we are going to discuss M phase, that is mitosis. In our previous session, I have sent you the video in which I explained about the various phases of the mitosis, that is prophase, metaphase, anaphase, telophase, and then cytokinesis in a diagrammatic way. And today we are going to discuss those phases in detail, what changes takes place step by step while the division of the cell into two daughter cells. Discovery of mitosis. So mitosis was described by German biologist Edward Strasberger in plant cell, whereas Walther Fleming described the mitosis in animal cells in 1882. Occurrence. Mitosis takes place in somatic cells in the animal. The somatic cells are known as the non-reproductive cells, which helps in the growth and the development of the animals. Whereas in the plant cells, it takes place in meristematic tissues, which has power of division, and during the growth of the leaves, flowers, and fruit. Duration. Mitosis lasts on average from 30 minutes to 3 hours. What is mitosis? How you will define mitosis? Mitosis is the division of parent cell into two identical daughter cells, each with a nucleus having the same amount of DNA, same number and the kind of chromosomes, and resemble exactly to their parents. So what is mitosis? It is the division in which two daughter cells are formed with the same number of the chromosome as that their parents. So it is known as equational division. Why? Because the number of the chromosomes in the parent cell and the daughter cell is the same. Phases of mitosis. It involves two phases. Karyokinesis, in which the division of nucleus takes place. Cytokinesis, division of cytoplasm. So first we are going to discuss karyokinesis, that is the process of division of nucleus, and it is completed in four stages: prophase, metaphase, anaphase, and telophase. And one thing very important, you have to learn these phases in this sequential order only. You cannot change the sequence of the phases, because in karyokinesis they takes place one after the another. So first we are taking prophase. It is the first stage in karyokinesis following the interphase. It is marked by initiation of condensation of chromatin material. Nuclear membrane, nucleolus start disappearing. In animal cell, centriole, because centrioles are present only in the animal cell and they are absent in the plant cell, that have been formed by duplication start moving towards the opposite pole of the cell. Condensation of chromatin material results in the formation of compact meiotic chromosome. Each chromosome is composed of two chromatids held together by the centromere. Initiation of meiotic spindle formation occur by the end of prophase. How it takes place? When the centrosome they start moving towards the opposite poles. By the end of prophase, nuclear membrane, nucleolus disappear, disintegrate completely. The Golgi complex, endoplasmic reticulum also disappear. So by the end of the prophase, we are just having a spindles and the esters in animal cells coming out from the centrioles and the compact meiotic chromosomes instead of the chromatin material. So in this picture you can see that is the condensed meiotic chromosomes attached by the centromeres with the help of a disc-like structure that is kinetochores, and these are the condensed chromosome, nuclear invert that start disappearing, and the centrosomes, they are separating, they are start moving towards the opposite poles, forming the meiotic spindles, and that rays coming out during the spindle, that is known as the microtubules. Next is your metaphase. This is very important phase. Condensation of chromosome is complete and chromosome are thickest and shortest in this phase. Spindle formation is complete. Kinetochores are small, disc-shaped structures at the surface of centromere that serve as the site of attachment of spindle fibers to the chromosomes that are moved into the position at the center of the spindle. All the chromosomes lie at the equator. One chromatid of each chromosome is attached by kinetochore to microtubules from one pole, while its sister chromatid is attached by its kinetochore to the microtubule of the opposite pole. What does it mean? So here you can see these are the condensed chromosomes with having a centromere in the center, and they are being supported by or covered by the disc-shaped structure that is kinetochore, which make them to be attached to the spindle fiber. These are the microtubules of one end and this is of another ends, by which they are held together. They are attached to the microtubules one on the other side, that is to the opposite side. The plane of alignment of centromere of all the chromosome at this stage is called metaphase plate. So here you can see that all the chromosomes, they are lying at one particular plate with centromere in the center, which are held by the microtubule, and they are lying on one plate. This one plate is known as equator or metaphase plate, and by this stage you can count the number of the chromosomes. So this is the only phase where you can count the number of the chromosomes. The next is your anaphase. In this phase you can see the spindle fibers, they are start breaking, they break, and centromeres of all the chromosome, they split simultaneously and moving towards the opposite pole, forming the daughter nuclei. Half of the centromere along with one chromatid is now called as the daughter chromosomes. As the spindle fiber contract, the daughter chromosome begin their migration towards the two opposite poles, and during this movement the centromere of each chromosome towards the pole leading edge wherever they are going, while the arms of the chromosome, they trail behind. So they are moving towards the pole, and arms, they are towards the chromosomes, they trail behind. So they are towards the end and they are towards the trail. So that is behind and that is towards the pole. Fine. So chromosomes are moving towards the pole and arms are lagging behind, or you can say the trail behind. Next is your telophase. It is the last phase of nuclear division in mitosis and considered to be opposite to the prophase. Whatever the changes is taken place in the prophase, they are revert back in the telophase. The chromosome have reached their respective poles and the spindle has disappeared completely. The chromosomes start their decondensation. In the prophase, we said the chromatid material condensed to form the chromosome. Here the chromosomes, they start decondensation and form the chromatin material. So they start their decondensation and become thin and long, and they lose their identity and form the network. Nuclear membrane reappear around the chromosomes cluster, that is the network. Nucleolus, Golgi complex, endoplasmic reticulum, they all reappear. So this is just the opposite to the prophase. In the prophase, the condensation of chromatin material takes place to form the chromosome. Here the chromosome, they decondense to form the network. Centromere, centriole, they form the spindle which disappears. Nuclear membrane disappear, it reappears in telophase. Nucleolus, Golgi complex, endoplasmic reticulum, they all get reappear in the telophase. So in this picture you can see they are at the two ends, that is nuclei, and then the two daughter cells are being formed with the same number of the chromosomes, and they are having a centriole, they are having a nuclear membrane, they are having a chromatin material. So whatever the things that is being disappeared in the prophase, they reappear in the telophase, and two daughter cells are being formed. Now next phase of your mitosis is cytokinesis. It is the process that accomplishes the division of cytoplasm and thereby cell into two daughter cells, each enclosing one daughter nucleus formed after karyokinesis. The process differs in animal and plant cells. In animal cell, furrow appears in the cell membrane during the late anaphase and early telophase. The furrow deepens, and when they join in the center, the cytoplasm, the cell is divided into two compartments. Each compartment with daughter nucleus is known as daughter cell with the same number of the chromosomes. Whereas in the plant cell, cytokinesis starts with the formation of precursor called as cell plate, which actually represents the middle lamella between the cell walls of the adjacent cells. This extends from the center of the cell towards the lateral walls and cuts the cell into two halves, each with the daughter nucleus. During cytokinesis, the cell organelle like mitochondria, plastid also get distributed between the daughter cells. So this is about the plant cell in which the cell plate is formed because that represents the middle lamina along with the cell wall, as the cell wall is absent in the animal cell. So that's why they are different in animal cell as well as in the plant cell. And there is one note over there that if cytokinesis does not occur, means the cell is not being formed, and karyokinesis is following, means from 2 nuclei, 4 nuclei being formed, then 4 to 8, then that is called as a multicellular condition, or syntium arises, means when more than one nuclei is being present in one cell, that condition is known as a multicellular condition, or syntium is there. So in this picture you can see this is about the animal cell in which the furrow takes place, that deepens out, and ultimately they form a two daughter cells. Whereas in the plant cell we are having a cell plate, they joins to the plasma membrane to form the middle lemma, then distribution of organelles takes place and two daughter cells are being formed. So in this image you can have a summary or the recap of all the stages of the mitosis. That is, in the interphase, nucleolus, nuclear envelope, that is nuclear membrane, are distinct, means very clear, and chromosomes are in the thread-like structure, that is your chromatid. Fine. Whereas in prophase, the chromosomes appear condensed, nuclear membrane, they start disappearing, centriole, they start moving towards the opposite pole to form the spindle fibers. In metaphase, they become thick coiled chromosomes with two chromatids, the centromere in the center, and they all are arranged in one plate, which is called as your metaphase plate. And in anaphase, the chromatids of each the chromosomes, they separated and they start moving towards the opposite pole with the splitting of the centromere. Chromosomes are towards the poles, and whereas the arms are towards the center. So the telophase, the chromosomes are at the poles and they're becoming more diffuse. Nuclear enveloped reforms, chromatin materials, they are going to be reforms, and all the organelles, they reappear. Then the division of the cytoplasm takes place, which starts with the early telophase, and by the end of the cytokinesis, the two daughter cells are being formed with the same number of the chromosomes and the chromatin material with the centriole, with the nuclear, so having the same amount as well as the same number of the chromosomes. So that's why that is called as an equational division. Next, and the very important, is the significance of mitosis. So what is the significance? As we have already discussed, that is takes place in the somatic cell, in which the growth and the development takes place. So in multicellular organism like we, that is human being, the body growth is by the meiotic division of the cell. Next, replacement of worn out tissues, cells, for example blood cells, skin cells, and repair of injured tissue is by the mitosis. So whenever we have some injury, the wound healing is by the process of mitosis. In unicellular organism, mitosis are involved in asexual reproduction. As you must be knowing in your previous classes, that is amoeba divides and two daughter amoebas are being formed, and amoeba is a unicellular organism, and they shows the mode of reproduction, that is the asexual, in which the same number of the chromosome, same daughter cells are being formed. So asexual reproduction means the mitotic division. Whereas in plants, vegetative propagation involves only the meiotic division, and genetically identical individuals are produced. So whenever the reproduction in the plant takes place by the vegetative parts, that is roots, stems of leaves, they shows the meiotic division and they are genetically identical with the plants. Uncontrolled cells in certain tissues or the organ, that is cancer, they results in the tumors. So uncontrolled growth of the cell, wherever they are not being required, they form the tumors. So that is again because of the mitosis. So this is all about the very important part of our cell division, that is M phase, that is mitosis, which helps in growth and the development of the body, and that is divided into two phases. Karyokinesis complete in four stages, that is your prophase, metaphase, anaphase, telophase, and you have to learn all the particular conditions of particular changes, events takes place in these phases in the sequence manner. You cannot change the sequence because they takes place one after the another. एक सीक्वेंस होगा, उसके बाद दूसरा सीक्वेंस उसे फॉलो करेगा. आप सीक्वेंस को बदल नहीं सकते हैं. यू हैव टू फॉलो दैट सीक्वेंस ओनली. प्रोफेस में सबसे पहले क्रोमोसोम्स आपके बनेंगे विद द कंडेंसेशन ऑफ क्रोमेटिन मटेरियल, followed by the disappearing of the nuclear membrane. फिर centriole, they start moving towards the opposite pole, spindle fibers बनेंगे. तो one after another the changes takes place. मेटाफेज में पहले स्पिंडल फाइबर्स पूरे होंगे, फिर क्रोमोसोम उसके ऊपर अरेंज्ड करेंगे. पहले आप क्रोमोसोम अरेंज नहीं कर सकते हैं, पहले आपको स्पिंडल फाइबर के बारे में ही बताना है कि वो कंप्लीट होगा. एनाफेज में पहले सेंट्रोमियर स्प्लिट होगा, फाइबर्स डिसअपियर होंगे, कॉन्ट्रैक्ट होंगे, तभी वो पोल्स की तरफ मूव करना शुरू करेंगे. When they are reaching to the pole, only then the recondensation or the decondensation of the chromosome takes place, then reappearing of the membrane will be there. You cannot write in telophase कि पहले nuclear membrane आ गया, नहीं, पहले chromatin material बनेगा, फिर उसका membrane बनेगा. So you have to learn all the changes, all the events in the sequence manner. I hope that is being clear to you. This is very important question. It may comes with a 5-mark question that you have to explain all the stages of the mitosis with diagrams. The diagrams are very important. So I try my level best to dictate it out, to give you the complete notes with the diagram, and you make you stepwise how to make a simple diagrams in your exams in the last video. So next lesson will be on our meiosis, which is the second cell division which takes place in the reproductive cells. So till then, stay home, stay happy, stay healthy. Goodbye. Thank you.